I'm pleased to be presenting today at this symposium and want to thank Lida for all of her hard work in getting it off the ground. So the first new American research with psychedelics began with our work at UNM in 1990 and wrapped up in 1995. And that initiated the renewal of studies across the country with a number of different compounds and a number of different settings. So just a brief background on who I was before I came to UNM. I started as a chemistry major because as a teenager I liked making fireworks and bombs. In college, my first cannabis experience was completely psychedelic. And with my interest in chemistry, I began wondering about how the psychedelic state is produced by changes in chemistry. This was the early 1970s, and meditation was also coming into the West Coast. And I was impressed or struck by the overlap in descriptions of effects of certain types of meditation, as well as those brought on by psychedelics, the visions and the voices, the emotions, the insights. So that began coalescing into the question of what is the biology of spiritual experience, or naturally altered states of consciousness. But my initial interest was began with spiritual experiences from meditation. The pineal gland was a rather obscure topic in human physiology at that point, but there was a lot of esoteric historical ideas about the role of the pineal gland in spiritual development. So I started wondering if there might be some psychedelic component in the pineal gland. So at a certain point I decided, well, I wanted to do psychedelic drug research, but this was 1971, 1972, and there was no one that was doing clinical work at that time. So I thought I would keep it in mind, and in the meantime get as well trained as I could, so if one day people could study psychedelics again, that I would have the proper training and background. So off to medical school, then a psychiatry residency at UC Davis, which was oriented towards community mental health, learning how to do psychoanalytic psychotherapy and prescribe medication. My first job out of residency was at a community mental health clinic in Fairbanks, Alaska in the early 80s. And there was a great case of synchronicity. This was around the time that winter depression was being described. And part of the mechanism behind winter depression was thought to be melatonin and the pineal gland. Melatonin is the primary hormonal product of the pineal. So all of a sudden, psychiatry is interested in the pineal, and I was interested in it for slightly different reasons. But it seemed like my opportunity to see about doing research and start off with the pineal gland. So I packed up from Fairbanks and moved to San Diego, where I took a fellowship at UC San Diego in clinical psychopharmacology research. And it was a very exciting time. I learned how to give things and to draw blood samples, give questionnaires, scrutinize scientific papers, do sleep studies. It was a very interesting time. And one of the things that occurred there is I contributed a chapter to a book on adverse effects of drugs, and I chose to do my chapter on psychedelics. And so by the time I began applying to do my studies, I had reviewed all of the effect. I had reviewed all of the literature from the first wave of interest in these drugs regarding adverse effects. And the consensus was if you screened people carefully, monitored their experiences, and followed up, chances of adverse effects were rather low. And since uh, the research model would require those things, screening, supervision, and follow-up, that this would be a good argument in support of any request to study psychedelics. So one of the people that I was in San Diego with was John Lasansky. This is kind of a new picture. I don't have one from our younger days together, but we were both in San Diego at the time. And he had done his residencies in internal medicine and psychiatry at UNM, and then came out to San Diego. And he returned to UNM, started working at the VA and doing clinical research there. And about a year after we finished up at San Diego, he invited me out to UNM and interview for potentially working there. He told me I would be able to do research, which wasn't the case at the position I was in at the time at UC Davis, having returned as a faculty member. So I came out to Albuquerque, saw John, met Walter Winslow, who was chairman of psychiatry at the time. And Winslow was interested in developing a nucleus of young researchers. And at this point, it was John. And with my interest in joining, he was quite supportive. Another major draw was the GCRC, the General Clinical Research Center at UNM Hospital. It was an incredible operation. It was one of a few dozen that were funded across the country by the NIH. And the logistical support was incredible. Things were underused, and they were just really keen on getting new people on board doing clinical research. So the director of the clinical research unit at the time was Glenn Peake. This is a pretty dashing photo of him. So Glenn was a pediatric neuroendocrinologist and was interested in melatonin because of its endocrine effects, potential role in puberty. And that's all that was really known about melatonin at the time. And he was quite supportive in bringing me on board to study melatonin in humans, the endocrine effects in particular, but other ones as well. Another great aspect of the research unit was Dr. Qualls, Clifford Qualls, a biostatistician who we spent endless hours crunching data for years. So the first big study, well, the first independent study that I did at UNM was looking at melatonin. And it was a post-fellowship training grant funded by the National Institutes of Health. From that research, which went on for maybe 18 months or so, we got 5 peer-reviewed papers, and in one of them we described the first known function of melatonin in humans, which was the regulation of the trough, the bottoming out of body temperature at 3 a.m. One thing we found out, which was of the greatest interest to me, was that despite some suggestions of a psychedelic effect of melatonin, there weren't any in our study. It was only sedating. So around that time, it was a few months after that paper on adverse effects came out. I got a call from Rick Doblin. This was like December 1984, and he saw my paper on adverse effects and wanted to invite me to an invitational meeting at the Esalen Institute that next year. So I went to that meeting a couple of times, maybe 3 years, 4 years, and it was a great place to network with other psychedelic types, especially people doing work on the underground who are nevertheless quite accomplished, either with the chemistry, the psychotherapy, the understanding of the phenomenon. So that was a great connection early on. And as most of you know, Rick went on to get his doctorate in public policy from the Kennedy School at Harvard, and is the mastermind, the mover and the shaker of MAPS, which has been so instrumental in getting MDMA close to approval as a treatment for PTSD. So I finished the melatonin study, and melatonin was not psychedelic. So I learned about DMT at the time, dimethyltryptamine, a chemical cousin of melatonin and serotonin, which is found in the natural world, quite widespread in the plant kingdom, in the animal kingdom, including humans. It's naturally produced. It is clearly psychedelic. It had been used in studies previously, both in Hungary and in the US. It was a relatively obscure compound and wouldn't garner the same kind of attention as, say, LSD or psilocybin. And it was quite short acting, which I knew would be a useful trait on a research unit because of how noisy and unpredictable they could be. So once I started thinking of a DMT study, I was confronted immediately with the Controlled Substances Act of 1970. It placed psychedelics into Schedule 1, the most restrictive category, which is for substances which are highly abusable, even though that's not really the case with psychedelics. There's really not that much of psychedelic abuse out there. Uh, Clearly, you're incapacitated when you're intoxicated, uh, and one can define abuse as leading to incapacitation. It cannot be administered safely even under medical supervision. But that's really not the case because the adverse effects in studies during the first wave of research uh, were quite low. It was quite a safe proposition. And no known medical use, which also isn't quite true because there were a lot of promising leads, especially with addiction and end of life despair. One of my mentors at UNM was Eberhard Uhlenhuth, who had gotten, who was quite experienced, quite a senior psychiatrist. He had started off at the University of Chicago and knew a lot of the pioneers of human psychopharmacology, and also spent a lot of time at Johns Hopkins. And he came out to Albuquerque, for the weather. He was winding down. He liked New Mexico, and his wife, liked it out here as well. And we were talking one day, and he kind of asked me, what I wanted to be when I grow up. And I said, oh, I really want to study psychedelics. And he said, well, let me tell you a cautionary tale about psychedelic research back then. So Uhlenhuth was a site visitor of the NIMH that was coming to the University of Maryland to see about renewing their grant. They were still doing some LSD and some studies with DPT into the early 70s from previous research protocols, and they were requesting their funding be renewed. but it was not renewed. And Ulan told me it was because they got religion. They were convinced it was the mystical experience that was healing, and that it was no longer a matter of understanding the mechanism of action of how psychedelics work, but applying the mystical experience to as many conditions as possible. And that didn't vote well in their review at NIMH. And Ulunhut was key in helping me develop the questionnaire that we used for the study. So the question of getting religion, he really wanted me to stay into the straight and narrow psychopharmacology. He didn't want me to venture off into some of the more far out things that I was interested in as well, the spiritual part or characteristics of the psychedelics. So another key player, perhaps the key player in moving things along, was a psychiatrist that was at that time at UCLA, Danny Friedman. So Danny Friedman and Uhlenhuth were good friends from Chicago, and Friedman really was influential at that time, the president of APA, of ACNP, on every possible psychiatric organization. And he had friends and students everywhere, or former students in high places. And he was also on the grants committee for the Scottish Rite Foundation for Schizophrenia Research. And when we met, he suggested that I apply for that grant to get things started. And I got that grant. It was the first financial support for the DMT work. At a certain point, things were just not moving along at all with me getting permission to get DMT and to give it. And he just made a strategic phone call to Dr. Schuster, who was at that time the chief of NIDA, and said, help young Strassman. So things kind of loosened up after that. Things kind of broke open, and the federal government became more receptive. So the protocol was called 89001, just depending on the numbering of studies that come into the Human Research Review Committee. And I submitted the protocol, maybe 6 pages, the informed consent, in September of 1988. And let me move my screen a little bit away. Yeah, and more than 2 years later, we gave the first dose of DMT. So hold on a second, I'm going to take a little break. The study was fairly straightforward psychopharmacology, as Dr. Uhlenhuth recommended, and as Dr. Friedman did too. He advised us not to move into therapy first, but to work out just the psychopharmacological data and to establish the fact you could do this research again. So the arguments I put forward to justify the study was that the nature of DMT might help us understand psychosis. There might be some abnormal metabolism of DMT because it is endogenous. It also was the prototype of abusable tryptamines that were still in use, such as LSD, psilocybin. And there were also a lot of basic science data that had been accumulated since the passage of the Controlled Substances Act, which needed to be either confirmed or refuted compared to the human or extended. The notion of DMT being responsible for schizophrenia somehow or another led to some interesting work. For example, some research looking at an antibody to DMT to use as an antipsychotic agent. So the study was pretty straightforward psychopharmacology, phase one, give low, medium, and large doses of DMT to normal volunteers with previous psychedelic experience. We chose those with previous experience for a couple reasons. One was that they could give uh, more articulated descriptions of the state because they'd been there before with other psychedelics. And they could also make comparisons between DMT and other psychedelics, and they would be less likely uh, to panic under the influence of a powerful psychedelic. So we were going to characterize the biological and psychological effects. Back then, the main biological variables were neuroendocrine ones, which are reflections of serotonergic receptor activation, things like prolactin and cortisol. So we were going to draw blood samples to see the effect of DMT on those neuroendocrine parameters. Also develop a new questionnaire to quantify the state and to develop a way to measure DMT in the blood. So it was interesting. We had 2 grants before one dose of DMT was given. But I was fairly explicit in my requests for funding for these first 2 grants by just saying that the first year or 2 may just be paperwork and trying to obtain the DMT and permission to use it. And that turned out to be the case. It was a 2-year project. So the person who helped develop the assay for DMT was David Shade, a great guy, super expertise in the lab. Someone else who helped with the questionnaire development was Bob Kellner, who just lived and breathed rating scales and was a great psychotherapy teacher and diagnostician. So there were quite a few hurdles during that 2-year period. Within the state, the study needed to go through the IRB, the Human Research Review Committee. And thankfully, at the time, 1988, 1989, the chairman of the IRB was Alan Frank, a psychiatrist at UNM, and a libertarian at heart. And one day we were talking, he looked me straight in the eye and said, "We do not play God on this committee." So his approach was, if it made sense scientifically and if the informed consent document was thorough and was honest, that there was no reason to hold this research back for political reasons. I also had to go through the scientific advisory committee for the CRC, and uh, I was the chairman of that committee at the time. So I excused myself, but still it was a good time uh, with them as well. And the New Mexico Board of Pharmacy. Uh, so uh, they gave us a Schedule 1 permit at the state level, but that was dependent on getting one from the DEA at the federal level. So things really slowed down once they were being discussed in Washington. The 2 main, or the 2 uh, uh, well, the 2 bureaucracies with which I had to deal at that time were the FDA and the DEA. The FDA is responsible for the clinical part of things, and they uh, will grant an investigational new drug permit to studies that they think are sound and safe. And at the end of all of this work, I ended up getting a IND for DMT, psilocybin, and LSD. And at one point, we had all of those in the inpatient pharmacy vault. Um, the um, the DEA are responsible for security, safety, theft, diversion, those kinds of things. So they're the ones that will give you the Schedule 1 permit. And we got one for DMT ultimately. And a few years down the road, we got import permits from the DEA to bring in psilocybin and LSD from Switzerland, which we did. So the main person at FDA was Curtis Wright, very nice guy. It took a year to find him, maybe 14 months. So one of the huge questions was, where would we get our DMT from? Who would make it? You know, who could, make super high grade DMT, plus who's got the permission from the DEA to make DMT? And it turned out to be Dave Nichols from Purdue. He's currently at UNC, University of North Carolina, working on LSD binding. So once we got the DMT, we needed to put it through quality control that the FDA required. That was a bunch of laboratory tests. And we also needed a kind of a crude analysis of DMT levels in the vials. So the concentration was very high compared to what would be in the blood. And we didn't really need the same kind of technology as in the lab. So toxicology people at University Hospital took that on. And the Office of the Medical Examiner also played a role in things like TB testing, pyrogenicity in the rabbit. I mean, it was a lot of requirements. So, so once the study began, I thought to myself, what happens if I never publish any real data? What if I'm hit by a bus before that? So I just kind of spelled out the process in this paper I wrote in 1990 when it came out. It was just a step-by-step description of exactly, what I did, what problems I ran into, and the way that I solved them. Dr. Uhlenhut, you kind of, it chided me at the time, more or less, saying, you've got the secret sauce, keep it to yourself. But still, I thought it would, that it would be a good thing if other people knew, because that would mean there would be more research. I got a couple of grants from NIDA. The first was a small grant for 2 years. And then after that grant was finished, I got an R01. They were funded first time around, which is an unusual thing, especially for a young investigator. And I think it was because the government wanted to study these drugs in humans. They just were kind of waiting, for a good study to come through. So the small grant was the first couple of years, the dose response work. The R01 there were a couple of questions. One was whether we could develop tolerance to closely spaced repeated dosing of DMT. Because if you take LSD every day for 3 days, 4 days, you stop responding. The same with psilocybin. But with DMT, that had not been established, and I thought maybe it was the spacing. And so we gave DMT 4 times in the morning. The intervals were 30 minutes, and there was no tolerance, which was very interesting. Also, they wanted us to start figuring out what receptor subtypes of the serotonin receptor were involved. And we blockaded the 1A with Pindalol, and it augmented the effects of DMT, which was opposite what you'd expect from the animal data. and we began some work giving psilocybin. So the DMT study, the first study, the dose response study ended in 91 or so, but it didn't appear in the archives of general psychiatry until 94. Um, and so there were two companion papers. The first was, the biological effects, and the next was a description of the psychological effects and the questionnaire. That questionnaire, actually, the HRS, we didn't. It has not been revised since 1994. It's still being used. Actually, it's been appeared in maybe 50 peer-reviewed manuscripts, journal articles. It's not as popular as, let's say, the mystical experience or the 5D ASC questionnaires. Those are the two most commonly used. There was a paper in biological psychiatry about tolerance, and a non-peer-reviewed paper came out discussing the Pindole 1A antagonism augmenting DMT's effects. So not many papers, fewer papers than the melatonin work. So I think one of the major concerns we need to be aware of is adverse effects, and that's going to they're going to start accumulating, and some may be really pretty dramatic, and the media may pick up on them. So I think it's good to be aware of adverse effects. The first wave of research may have been falsely reassuring when it came to reporting adverse effects. And there's some thought that there was, the investigators were afraid if they described the true incidents that would put that would slow down the research. So in our study, I'd say maybe 10% of our volunteers had some kind of problem. I mean, it was uncomfortable. So in two of the volunteers, there was a recurrence of depression. One responded quickly to meds, starting back up on the medication, which years before had been used and were effective in a depression. And one person responded to some psychotherapy that was picked up right away. One person had a really nightmarish experience, just, it's X-rated. One of the volunteers developed panic attacks, but was able to work through them on his own and participated in the study fine. So, they were not completely trivial, but they did respond to interventions to support the referrals. There was one case of severe hypertension, one case of severe hypotension. We started giving people psilocybin. It's interesting, too, regarding the dose. The point at which the volunteers unanimously agreed that they were now under the influence of a psychedelic drug, that it was now a psychedelic experience, was 0.45 milligrams per kilogram given orally. And a handful of volunteers came in and received a slightly higher dose, 0.7, and the consensus was they could do more. So we gave two volunteers 1.1 milligram per kilogram, which was too much. So if we had gone ahead with the full dose response study, our high dose would have been 0.9. And compare that with the current high doses being used in psychotherapy, brain imaging studies. It's 0.45, which is perhaps just the threshold. I've never been able to make a sense of that comparison. So even though we answered a lot of biological questions, the undercurrent, the underlying spiritual question, which I brought to bear on this study, was whether DMT is inherently if it's inherently spiritual. In other words, if you don't give a lot of preparation and encouragement and steering of the experience, if you just support people, is it going to be spiritual? And I was practicing Buddhism at the time, and uh, uh, it brought the idea of Kensho and emptiness and enlightenment into the equation. Uh, but instead, the effects were psychedelic. They manifested or disclosed the contents of the person's mind. In other words, in a relatively unstructured setting, the people bring their own personalities to the experience, and that's what the experience builds upon. So one way of thinking about it is as a non-specific amplifier of more or less conscious aspects of the pre-existing personality. And one way of expressing that is to uh, you call the effects uh, meaning enhancement. Things were the case before, they weren't quite the case before, but under the influence of the drug, they becoming true, meaningful, much more important. So the psychotherapeutic and ultimately the spiritual effects um, were of interest to me, and I realized that there'd be a lot of work involved in the setting to do therapy or spiritual work. And I was unable to recruit people. It was just maybe too far ahead of the game or of the current or of the wave. Um, it was just the beginning of studies, and most people really hadn't thought through what they would do if they were actually able to give psychedelics. Um, so we ran the study from 90 to 95. We gave a lot of DMT to a lot of volunteers and a small number of psilocybin doses. And I think just as importantly, we established a protocol for Schedule 1 studies. So I left UNM in 95. I couldn't break out of the constraints of soul, solely psychopharmacology. I was interested in branching out. So I moved to BC, to British Columbia, to help with my family at the time. Moved to Washington and back to New Mexico, where I spent over 10 years practicing community and private psychiatry. And then in 2001, the Spirit Molecule book came out, and the commentary, the documentary a few years later. So our study, more or less, began American human research. It also influenced Rick Doblin because it served as the case for the development of his model, for forming and um, doing work, for maps. Our work established that you could study psychedelics, and Doblin pointed to it and said, if you can, then this is what I'd like to do. Mostly though, it's been consultation, providing advice, copies of the NIH-funded grants, the data, informed consent. There are a couple of MDMA studies that built upon my work, and I helped them in executing it. There was a University of Arizona study giving psilocybin for OCD, a Miami study giving ibogaine to normals, the Johns Hopkins mystical experience in normals using psilocybin. Um, when Michael Bogenschutz was in the department, I provided some information about psilocybin in developing the study of psychotherapy in alcoholism. There's a uh, paper came out last year, within the last 6 months, a Yale IV-DMT study for depression. I consulted with them, and Imperial College in London has been giving both continuous infusions of DMT, and that's building upon work that they did with single doses. So what's the big picture? Well, the big picture is that psychedelics do everything. I mean, look at this list. Either there's peer-reviewed papers demonstrating it, there's survey studies demonstrating these findings, um, the popular literature and press, uh, suggestions for conditions that would respond. Um, yeah, depression, end of life despair, the list goes on. I didn't count those up, but I should have. So if you think of psychedelics as super placebos, they'll basically um, direct the placebo response in the direction that you want it to take. Uh, we'll talk about that some more. Uh, and there's a lot of debate about what kind of subjective effects are required for the desired outcome. There's quite a few questionnaires. The most commonly used is the MEQ, the Mystical Experience Questionnaire, which scores on that seem to correspond to improvement in various psychiatric conditions. But I think other questionnaires would also demonstrate comparable findings once they're developed. Um, but I think it may turn out to be the case as the intensity of the experience, as opposed to the flavor, as it were. If it's a strong enough experience, that the intensity is what correlates with outcome, and it can be any number of experiences or types of subjective effects. There's a lot of controversy nowadays about can you take the can you remove psychedelic effects from the psychedelics and you still get benefit. And people are pointing to neurogenesis, the production of new neurons from stem cells, and neuroplasticity increases the complexity of functional uh, synapses among dendrites. Psychedelics will stimulate both neurogenesis and uh, neuroplasticity, and you don't need to give uh, behaviorally active doses in lower animals. That's not been studied in humans, but in lower animals, you can see uh, these neurological effects without any subjective or behavioral effect. So that's leading to thoughts about uh, neurological care for stroke rehab, neurodegenerative disorders, inflammation in particular. Um, or you could give sub-behaviorally active doses of the classical compounds, sub-behavioral effects of DMT in a lower animal, and there still will be increases in neuroplasticity. So there are brand new compounds being developed which are not behaviorally active, which are neuroplastogenic, and small doses of the classical compounds. So the non-psychedelic ones, I think, may turn out to be like super antidepressants, and they'll be a lot more effective if combined with psychotherapy, just as in the case of psychologically active compounds. And a lot of people will still either need or want a psychedelic experience. Recreationally, spiritually, you want a trip, and you might be called upon to use a psychedelic experience in a particularly difficult case. It's potentially greater benefit, but also potentially greater risk, and you have to do a risk benefit thinking through an analysis. So I think we need to keep a lid on our enthusiasm, or curb our enthusiasm. There's a lot of promising data. There's more hype and hope than uh, experimental, objective, data, to pull from. Things are promising, but we don't want to glorify the potential benefits or minimize the risk. There's a lot of risks that are incumbent, or that are associated with uncontrolled psychedelic use, recreational in kids, and mentally unstable. So these include suicide, psychosis, flashbacks, all the things that we read about from the 60s, early 70s. We want to be on the alert for psychedelics being blamed or finger pointed at for political upheaval and rebellion, kind of like what happened in the 60s with the conflation of LSD and the anti-Vietnam movement. And you be prepared for malignant uses. There's going to be another Charles Manson. There'll be terrorists, ultra nationalist groups, and the like that are going uh, to be taking psychedelics because of their effects on enhancing meaning, making things more true, and that it can go both ways. So we ought to be circumspect and hopeful at the same time regarding the future of psychedelic studies.